Welcome back, my biologists. So we are moving into chapter 7 now, and we're going to talk about membranes. We're going to talk about phospholipids, we're going to talk about proteins, we're going to talk all the stuff that's going on inside of a membrane, what it's made of, how it works. We're mainly going to talk about the cell membrane of a mammal cell, but we will also, these same concepts apply to the cell membranes of other types of organisms and also the membranes around your organelles, like your mitochondria. They all have the same kind of basic structure and chemistry that goes to them. A cell membrane is what defines the boundaries of a cell. It determines what is in the cell and what is out of the cell. And the membrane of an organelle determines the boundaries of that compartment. It is going to play multiple different roles over the life of a cell. It's not just a fixed gate that doesn't do anything. It's actually a very active player in the activities of that cell. The plasma membrane refers to the membrane that goes around the whole cell. So here, this would be the plasma membrane going around, or the membrane going around the cell. And then the intracellular membranes go around the different compartments. So here's a nucleus, and you have the nuclear membrane going around it. Here is a chloroplast. You have a membrane going around the chloroplast. So we have different membranes surrounding the different compartments of a eukaryotic cell and also around the entire cell. The different roles of a membrane include: 1, to define the boundaries, and to act as a permeability barrier. So if you remember when we're talking about those cell membranes, they're made of phospholipids, which give it a hydrophobic ring around the cell. So that is a pretty good barrier. Water outside the cell can't just rush in. Water inside the cell can't just rush out because it's blocked by that hydrophobic barrier of the cell membrane. The membrane is a site of biological functions. So in a mitochondrial membrane, you'll have the proteins for electron transport to be able to get energy from your cell. Or in a prokaryotic cell, you'd have those equivalent functions happening in the plasma membrane. Protein processing happens in the membrane of the endoplasmic reticulum as those ribosomes attach to the membrane of the ER and are creating proteins. Different proteins, transport proteins, are going to regulate the movement of things in and out of the cell, in and out of the organelles. So while we do have that hydrophobic barrier, you are able to send things in and out of the cell through these different transport proteins. You can bring in nutrients, you can send out waste, you can also keep a constant levels of different ions. We'll particularly talk about sodium and potassium in a couple of chapters. Some of the roles of membranes are going to be for signal detection. So like if a cell needs to grow and that growth factor comes, maybe this blue dot is a growth factor, attaches to a receptor that tells the cell it's time to go through the growth cycle, or a hormone coming and telling the muscle cell to release glycogen. And also cell-to-cell interactions. So the way that two cells interact with each other is going to be, a lot of that will be controlled through the membranes, how they're able to adhere to each other, how they're able to communicate with each other, to sometimes even share materials from one cytoplasm to the next through materials in the cell membrane. The reason membranes have these different jobs is going to be because the proteins that carry out those jobs are embedded in the membranes. So it is able to control transport because it has transport proteins. It's able to do signal detection because it has molecules for signal detection. It's able to interact with other cells because it has the molecules to interact with other cells. Those membranes that do those jobs are contained and embedded within the membranes. There are different membranes that have specific enzymes with them, and so you could stop and actually characterize what membrane you're looking at by seeing what enzymes are there. So if you were to isolate a mitochondrial membrane, you'd find different enzymes than if you were to isolate a membrane of the Golgi apparatus. So those are able to characterize that membrane. Transport of solids is controlled through the proteins that are in the membrane. So the membrane proteins will carry out and regulate the transport of substances, bringing it in and out of the cell. The cell will bring in nutrients. The cell will bring in ions. The cell will bring in gases. The cell will bring in water. All the things that that cell needs to keep it healthy and functional, the cell is going to bring in through those transport proteins. And as the cell is growing and doing its tasks, it will create waste that gets sent out through transport proteins. It'll also produce particular products. So we just talked about, if a cell is signaled to release glycogen, it'll break that glycogen down into glucose molecules that can be released into the blood. Or if it is creating a hormone, the hormone can be released out into the bloodstream to travel through the body. Those products get released through proteins that are in the plasma membrane. Some things are going to be able to diffuse straight across the membrane, things that are small, things that are nonpolar, uncharged, things like oxygen and carbon dioxide. They can typically just diffuse right through that plasma membrane and reach an equilibrium between what is in and out of the cell. Some things are going to require specific transporters. So here you see these transport proteins. Sodium ions and potassium ions have a charge to them, so they can't diffuse to the membrane, so they go through these membrane proteins. Nutrients might be a little too big, or they might be a little polar, so they won't be able to go straight through that plasma membrane. They also will need to go through a specific transport protein. The membrane proteins will detect electrical signals and chemical signals. So it can detect charges, negative or positive charges, and it can detect different proteins. It can detect different sugars or different hormones. And when a specific molecule binds to its receptor, a specific signal binds to its receptor, it starts what's called signal transduction. Signal transduction is what happens inside the cell after the signal is received. So the signal binds to the outside of the signal detection molecules, and that'll start a signal of, you might start phosphorylating, or it might start changing shapes or generating activities. It could even include ending up with a molecule that goes into the nucleus to act as a transcription factor to turn on genes or turn off genes. We discussed how different growth factors can start signal pathways through AKT or through RAS and RAF. So that is a signal transduction pathway where the binding of something outside the cell gets transmitted to inside the cell, which then sets off a pathway to change transcription of genes. I think this is just more of what I just said. When a signal molecule binds to the receptor, that triggers chemical events on the inner membrane surface that eventually are going to lead to other things, could even go as far as transcribing new genes. Membrane receptors allow cells to recognize, to transmit, and to respond to different signals. This plays an important role in all of our cells being able to interact with each other and form functional tissues, and for tissues to interact together and form functional organs, and even to have a functional entire organism. Membrane proteins also are going to be responsible for cell adhesion, or having two neighboring cells stick to each other, which is key to having a multicellular organism, and also for cell-to-cell communication, so the different cells in your body can coordinate their activities. So in our body, as people, we are multicellular. Our cells are pretty much always going to be in contact with the other cells. And there's 3 different kinds of junctions that we specifically have in animal cells. There are adhesive junctions. Adhesive junctions are these junctions that hold cells together. They adhere. And that's mainly because of cadherins. There are tight junctions that are going to form tight seals. So think of like the cells lining your intestinal tract. You don't want your partially digested food leaking out of that intestinal tract, leaking into your body. So those tight junctions keep those cells tightly sealed together. Or even in your bloodstream, you don't want your blood leaking out when it's not supposed to. So we have junctions that hold those cells together. And gap junctions are going to be things that actually allow little gaps between cells. So these are junctions that allow the things in the cytoplasm of this cell to travel to the cytoplasm of this cell. So it's helpful for them to be able to communicate. In animals, we have gap junctions, but in plants, they have plasmodesmata, and plasmodesmata perform a similar role. They allow nutrients or signals from one cell to travel into the cell beside it. They're little gaps. Okay, I'm sure you've heard of the fluid mosaic model. This is something you should have been very comfortable discussing in your first semester biology classes. The fluid mosaic model is the model that we use to describe all of our biological membranes. Your cell membranes, your organelle membranes, they are fluid because they are moving around, and it's a mosaic because it's made of different kinds of stuff. We have phospholipids, we have proteins, and even the proteins, we have different kinds of proteins. In this model, we have the membrane as two layers. So if you look here, you can see here's the phospholipids and the little lipid tails forming one layer. And then here's the phosphate heads and the little lipid tails forming the second layer. So it's a bilayer, it's two layers. The phospholipids can move around, they can twirl, they can move from here to here. The proteins can kind of open up or close up if they need to. They can wiggle a little bit. So it's fluid. The lipids and proteins can move pretty much only laterally, moving around like this. We don't often see a lot of moving from, one side to flip-flopping around or turning a protein upside down. And it's a mosaic. So a mosaic refers to those art pieces that are made of like crushed tiles, and you put lots of little pieces together and form the final product. It's the same thing here in your cell membrane. You've got all these little pieces that together interact together, form a functional membrane. You've got glycoproteins, you've got glycolipids, you've got phospholipids, you've got channel proteins, you've got cholesterol, you've got membranes just on the periphery, you've got membranes that go, so you've got proteins just on the periphery, you've got proteins that go all the way through, we've got lots of little things that together create that final big picture. But the way that we got to the phospholipid bilayer fluid mosaic model, we didn't figure this out right away. So I want you to think back to the 1600s, Hooke or Leeuwenhoek, and I know that's not how you say his name, but I'm not Dutch. So Hooke saw cells for the first time. He was looking at cork cells. What he probably saw were things like the cell wall and the nucleus. He probably didn't actually see a membrane. Or Leeuwenhoek with his animalcules, he could see, things like amoebas or large bacteria, but he didn't see a membrane. At that time, membranes, even though we knew cells existed, membranes were still kind of theoretical at that time. There had to be something around them, but you couldn't see a membrane. 1800s, they still didn't have microscopes to be able to see a cell membrane, but Charles Overton had a very forward-thinking, just for the data he had a really good ability to take that data and make it make sense to what the cell was doing. So he noticed that lipid-soluble substances were able to penetrate into the cell really well. So he figured that there had to be some kind of lipid coat around the cell that was preventing hydrophilic things from traveling into the cell very easily, but hydrophobic things could get in there very easily. And Irving Langmuir dissolved phospholipids in benzene, and he found that they formed a monolayer in water. So he discovered they were amphipathic. Amphipathic means they had a hydrophilic end and a hydrophobic end. So from this, he figured out that they must orient on the water. So the hydrophobic tails would move away from the water, and the hydrophilic tails would move away from the water, and the hydrophobic heads would I still said that wrong. The hydrophilic heads would move against the water, and the hydrophobic tails would cluster away from the water. They came up with this idea of a lipid monolayer surrounding your cells, these phospholipids forming a layer around your cells. In 1925, we still do not have microscopes that can see the cell membrane, but Gorder and Grindle are taking lipids from red blood cells and spread them out as a monolayer. And they discovered there was twice as much surface area as it would actually take to cover those blood cells. So either they had done something wrong and had miscalculated the surface area of the cells, or there actually was enough phospholipids to form 2 layers on the cells. And so they came up with the idea of a lipid bilayer. So there are not just this 1 layer from Langmuir, but 2 layers of phospholipids, which then allow for heads that are hydrophilic with the water inside the cell and outside the cell, and your hydrophobic tails can cluster together and interact in their nice little hydrophobic shell without having to interact with water. Then Hugh Davison and James Danieli figured out that just having a bilayer of membranes, that totally fit the data, but there had to be more to it. It couldn't be just phospholipids. They needed to be things like proteins because of things that they were observing about surface tension or solute permeability, that things that were hydrophobic were still sometimes, things that were not hydrophobic were still sometimes able to get into and out of the cell. There was some electrical resistance. So there had to be proteins doing something in there. So they came up with this idea that you have a phospholipid bilayer, and then you just had a thin coating of proteins on either side of the bilayer. So some proteins covering out here and some proteins covering out here. Starting to get closer, but you're still not quite there. There's still some problems with the data that don't quite fit this. So one would be that there's not enough space for a protein coat. When scientists started measuring how much space there was between 2 neighboring cells, there was room for the phospholipids of a membrane, but not if you started adding proteins on either side of it. They also found that protein and lipid compositions vary. They also found something called phospholipids, phospholipase, that would dissolve the phospholipid heads. If you dissolve the phospholipid heads, you should take the proteins with you, but the proteins were not being removed with those phospholipid heads. They were staying where the lipid part was. And they also found proteins that had amphipathic properties or hydrophobic properties that just wouldn't have been able to very well function or keep their shape in this coating on either side of the membrane. Eventually, we had electron microscopes developed, and at this point, we could actually view the plasma membranes. So it wasn't until electron microscopy was available that we could actually physically verify that there was an actual plasma membrane around cells and around organelles. And when we started looking at the cells, we saw here's the first cell, here's its phospholipid membrane, then there's an intercellular space, and then there's the other phospholipid membrane and the second cell. This is called the trilaminar appearance because it looks like a layer, and a layer. That same trilaminar structure was observed in all membranes, whether you were looking at an animal or a plant or a bacteria or any kind of cells that were attached to each other. You have that same trilaminar structure. So this developed into the idea of the unit membrane. The unit membrane said that we have this common membrane structure. No matter what kind of cell you are, no matter what kind of organism you are, your membranes are going to be the same. Your membranes are the same in your cells of an animal. Your membranes are the same in the cells of a plant. Your membranes are the same in your organelles. That is our unit membrane. Membranes are this basic phospholipid protein combination structure. Then eventually, Singer and Nicholson introduced the idea of a fluid mosaic model. So this is going to take those data inconsistencies from our previous models and put them together. Fluid mosaic model has 2 key features. One is fluid. The lipid bilayer is a fluid. You have 2 layers of lipids, and they can move, they can twist, they can roll around to different spots of the cell membrane. And it is a mosaic. There are a mosaic of different proteins that are embedded in the bilayer or attached to the bilayer. So by embedded, I mean they're actually shoved into it, or just slightly attached. It might just be a little bit in one side. It might just be without even going into the membrane. It might just be attached just on the surface of it. This is a model that we use today, the fluid mosaic model. We understand that there are some integral membrane proteins, and they could have multiple hydrophobic segments, and each of those hydrophobic segments are going to span the lipid bilayer. These are called transmembrane segments. So here's an example of a protein that it starts here, it goes through the membrane, through the membrane, through the membrane, through the membrane, through the membrane. So it goes through the membrane multiple times. It has multiple transmembrane segments or transmembrane spanning segments. Bacteriorhodopsin was the first protein that we really studied to see how it was able to go through the membrane. Bacteriorhodopsin is a protein that is found in the cell membranes of archaea that is involved with some of those prokaryotic processes. And recent findings also suggest that membranes are not homogeneous. So even though we have this same kind of fluid mosaic model for all membranes, the cell membrane of different cells are going to vary some. They're not going to be all the same. And even within one cell, you will have particular areas that functions are grouped together. So you don't have the same even spread of proteins all around the membrane. They have specific organizations and groupings as different functions are connected to each other. Okay, we're going to come back. We're going to talk about in our next videos some more roles of the phospholipids within your cell and how they are controlling a lot of the functions of your cell membranes. All right, I'll see you soon. Bye.